So this time let's continue on with proteins. So with proteins, they're basically amino acid polymers, polypeptides, and they have peptide bonds which help to make them. So let's talk about their functions. Their main functions include structure, storage, transferred, defense, and enzymes. So one of the things is that there is such thing as storage proteins. This can include casein, milk, or alveolum in egg whites, or zein, which is corn. And we have transport proteins as well. So that could be hemoglobin because it transports oxygen, or cytochromes, which transport electrons. Finally, there are also enzymes, so they can catalyze reactions, forward or reverse direction. Remember, enzymes do not change the spontaneity of the reaction. The efficiency is based on the substrate, enzyme concentration, temperature, and the pH, along with the presence or absence of inhibitors. So, for example, amylase will catalyze reactions which break alpha glycosidic bonds in starch. There's also an exception. While enzymes are mostly proteins, there are also times where RNA can be an enzyme, which is called a ribozyme. Let's also just talk quickly about the structure of proteins. Not just the basic structures of an amino acid. So we can have H and H here. And then we have a central carbon, hydrogen, and then an R group. And then on this side, we can have C, O, H, O, a carboxyl group. So there's different parts to the amino acids. So we can have the carboxyl group. And then we have the amino group. Finally, we have the R group, which basically gives you the function of that specific amino acid. And we have an alpha carbon at the center, so your central carbon. Yeah, just good to remember the structure of the amino acids. So let's move on. Next, we have cofactors. Cofactors are non-protein molecules. Why are they part of proteins, though, in this section? Because they help assist enzymes in their actions. So if it's organic, it can be a coenzyme, such as vitamins. And if it's inorganic, they're basically metal ions. Finally, if the cofactor is covalently bonded to an enzyme, then it's referred to as a prosthetic group. And when it binds, cofactors bind to the enzyme, they are called holoenzymes, basically. Let's also talk about how you classify proteins. So you can classify them in two ways, simple or conjugated. Simple, it makes sense. They're entirely made of amino acids. But conjugated include of simple proteins along with some non-protein molecule, like a cofactor potentially, who knows. So simple. Simple consists of albumins and globulins. I hate pronouncing those, but that's what they consist of. And these function to carry enzymes. And we also have scleroproteins, which are fibrous proteins, and they have a structural function for collagen, for example. So those all are simple proteins. Conjugated, on the other hand, include lipoproteins, so a protein bound to a lipid, or mucoproteins, so a protein bound to a carbohydrate, or chromoprotein, which is protein bound to a pigmented molecule, or potentially a metalloprotein, so a protein complexed around a metal ion, and a nucleoprotein, so protein with a histone or a proteamine bound to a nucleic acid. Yeah. One other thing that's very important with proteins is discussing their structure, because their structure technically determines their function. So let's talk about the primary structure of the amino acid. The primary structure is just the linear sequence of the amino acid connected by peptide bonds. So usually if you were seeing a drawing along those lines, it'll just be a bunch of amino acids connected by peptide bonds in just a linear simple chain. How about the secondary structure? Well, the secondary structure would be 3D shape. It's hydrogen bonding of amino and carbonyl groups. So, for example, the alpha helices or the beta sheets. Those are both the secondary structure, or refer to those, of proteins. Let's talk about the tertiary structure. So this is the 3D structure folding of proteins. This includes the non-covalent amino acid R group interactions. So let's put these out. So say we have this huge protein. That might just represent the tertiary structure. This would include van der Waals forces, hydrogen bonds, ionic bonds, hydrophobic bonds, includes disulfide bonds, and a strong covalent between cysteines. So just remember that. Not cytosines, not to be confused with that, just cysteines. And finally, we have a quaternary structure. So this is the 3D shape of a protein with 2 or more polypeptide chains. So think of hemoglobin. There's more than one polypeptide chain there. So basically, if you see only one polypeptide chain, you can basically conclude that's the tertiary structure. Let's move on to the globular proteins. So globular. So globular proteins are soluble in H2O, and they have a dominant tertiary structure. Soluble. Dominant tertiary structure, and their function, they have such a wide variety of functions, including enzymatic, hormonal, inter- and intracellular storage, and transport, osmotic regulation, and immune response. We also have fibrous or structural proteins. So fibrous proteins are not soluble in water. They have a dominant secondary structure. Oops. They consist of long polymers, and their functions are to maintain and add strength to the cellular plus matrix structures. Finally, we also have membrane proteins. I won't write them down because we're going to discuss them later on in detail, but their functions are basically to be membrane pumps, channels, or receptors. Yeah. Finally, let's also talk about protein denaturation. So what does this consist of? So this is a reversal to the primary structure of a protein. The protein becomes non-functional at this point, and this could be due to temperature, pH, salt concentration, UV lights, and chemicals. Denaturation can occur for a variety of reasons. And once the protein returns to this primary structure, it no longer can function. Remember, do not get this mixed up with digestion. Digestion eliminates all protein structure, not just goes back to the primary. It just removes all the structure. All right, yeah, that was proteins. We're going to go on to nucleic acids after this.